So next up we have Raul Mostoslavsky. All right, from Harvard, and that looks good. All right, so his title is Linking Aging and Metabolism to Epigenetics and Cancer, One Gene at a Time. All right, take it away. Looks good.
Thanks, Ashley. Can you hear me well?
Yep, sound great.
Okay, so yeah, thank you. Thanks. I wanted to thank the organizers first, Peter Adams and all the team at Motif for inviting me to be here today. I wish I could be in person there in California. Last time I went to the West Coast, I was lucky to go to the Grand Canyon for one night, and I took this picture that you see here, which actually was selected for Photo of the Day, National Geographic, and got an award from Nature Photographer of the Year. So very proud of the picture. Hopefully, at the end of the meeting, I can tell you how I took it. But moving on to the science. So my laboratory has been very interested in understanding this crosstalk between metabolism and epigenetics for many years. And I call it here an obvious link. Although many people in the chromatin field don't think about metabolism, and vice versa, many people studying metabolism don't think about the epigenome. But I said an obvious link because, and you can see here a few examples, for most of the chromatin modifications that we have in our cells, you need specific metabolites, and they tend to be limiting. You cannot methylate histones or DNA if you don't have enough amounts of SAM, the S-adenosine methionine universal donor. The same is true for acetylation of histones. You need acetyl-CoA, a major nodal point in the mitochondrial metabolism, and actually work from many, including Shelley Berger, show nicely, and Cathy Wellen, that if you reduce levels of acetyl-CoA in hours, you see changes in histone acetylation. So availability of these metabolites are key for these epigenetic changes. And lastly, they are not only substrate for the reaction, they are also key cofactors for many chromatin factors. And a good example is NAD, one of the major molecules in the TCA cycle in the mitochondria that is used as a key cofactor by many chromatin enzymes, including PARPs and sirtuins. And my lab has been working on one of these sirtuins, SIR6. I don't think I need to introduce sirtuins in this audience. Originally described in yeast, they all use NAD as a cofactor, as I mentioned, and many of them extend lifespan. My laboratory has not been working on aging, but I believe I am being invited to this meeting because we've been working on one of these sirtuin called SIR6 in mammalian cells. It's one of the exclusively found primarily on chromatin. That's why our interest is coming from the epigenetics field. But interesting to say is that when in mammals, one of the main mammalian homologs that did show extension of lifespan is SIR6 itself. And recent work by Vera Gorbunova, who will be a speaker later in this meeting, showed that the activity of SIR6 is linked to longer living species. So as I said, we haven't been working on aging per se, but it's a very unique molecule to study. I won't have time to tell you about all the work that we've been doing in the lab, and we move away from only SIR6 in the past few years. We've been interested in understanding roles that is unique, the acetylase, and we've been publishing throughout the years major roles for SIR6 in regulation of tumorigenesis. And this will be one thing I will tell you about today. But we've been expanding, as I said, to other roles of metabolism in controlling changes in chromatin, particularly roles for one-carbon metabolism that I'm not going to tell you today. I keep my close interest in roles for chromatin in DNA repair, and we've been working on this in the lab also, but as I said, I won't have time to tell you about it today. And more recently, we became interested in unique adaptation of metastatic cells compared to primary tumors, and I hope I will have time to tell you one story related to these last studies. So the two stories I hope to tell you today, one relates to metabolic heterogeneity in cancer, and the second one relates to non-genetic drivers of metastatic disease. And for the first, it's based on our original work with SIR6. I won't show you the original data. This we published many years ago, but we discovered SIR6 as a key modulator of what we call a glycolytic switch. Most of the normal cells keep SIR6 sitting on genes on metabolic genes co-repressing a factor called HIF-1 alpha. And this is true in normal oxygen and nutrient conditions. By sitting there, it's repressed HIF, so you don't get expression of what are called glycolytic genes. And here are a few examples: pyruvate dehydrogenase kinase 1, lactate dehydrogenase, and the glucose transporter GLUT1. By keeping repressed those genes, most of the pyruvate that get into the cells go to the mitochondria to produce ATP through respiration. The most efficient way to produce ATP from pyruvate. Conditions of low oxygen or low nutrients, we found that CIR6 is kicked out from chromatin. You transactivate HIF-1 alpha, you bring co-transcriptional activators that they acetylate these genes. And by acetylating these genes, we get upregulation of these genes and a switch where the cells, instead of respiring actively, they keep respiration, but in much lower way, but they start producing much more lactate from pyruvate. Less efficient to produce ATP, but gives a cell an opportunity to survive until conditions improve. We showed this in normal cells many years ago, and it was one of the first examples of a metabolic switch that was controlled by a chromatin regulator. Now we realized that, and I'm sorry, a sidetrack story, one slide. When I originally created this figure, this animation, we put CIR6 at the promoter because most of the HDACs, histone deacetylases, repress transcription by sitting on the promoter, compacting chromatin, and inhibiting the recruitment of the polymerase. This original model was wrong. Work we did on ChIP-seq to see where CIR6 was sitting genome-wide and show us that different from instant for HDAC1. Here you have a HIPMA where you see binding of the gene, of the proteins on genes. Each lane is a gene, and you clearly see HDAC1 sitting at the TSS. You are looking here at the TSS, 3 kb upstream and downstream. And in contrast to other HDACs, we see CIR6 sitting intragenically. And long story short, what we found in this paper was that it's a very unique HDAC in the sense that it binds the polymerase, move intragenically, and control what is called transcriptionally pausing and elongation by regulating the recruitment of specific elongation factors that I will not have time to tell you today, but please refer you to the paper. And we believe it's a unique adaptation of this HDAC to regulate and fine-tune expression of metabolic genes. And we can discuss this at the end if we have time. But back to the metabolic story, what I didn't tell you is that by getting rid of this transcriptional co-repressor CIR6 and HDACs, we were eliminating the sensing of these changes in nutrient or oxygen. And our cells that didn't have CIR6 were constitutively producing lactate, regardless of the levels of oxygen or nutrients. And this is called aerobic glycolysis, and it's a feature that Otto Warburg described many years ago in cancer cells. And Warburg said, regardless of the amount of oxygen, cancer cells tend to produce lactate from glucose. He didn't understand exactly why, but this was exactly the phenotype we saw for these CIR6 deficient cells. So we hypothesized that maybe cancer cells get rid of CIR6 to acquire a metabolic adaptation. Now, for the past 10 to 15 years, many laboratories have been studying this Warburg effect in cancer, trying to understand why cancer cells do Warburg. And this is a very heavy slide. I'm not going to go through all the details. The only thing I wanted to mention here, that from the work of many laboratories, what we believe is happening is that during the process of converting pyruvate into lactate here, instead of sending pyruvate to the mitochondria, which is a much more efficient process, you accumulate intermediates. And these intermediates are used for side metabolic reactions, particularly the generation of nucleotides, RNA and DNA nucleotides through the pentose phosphate shunt, acetyl-CoA for lipid synthesis, and serine through glycerol 3-phosphate for amino acid synthesis. And the production of the three basically lead to biomass, to an increase in biomass, and rapidly proliferating cancer cells may benefit from doing Warburg by obtaining these bricks for biomass. At the same time, in every one of these step reactions, you produce reduced NADP, contributing to a major change in reductive power. So the field has been thinking that the major reason you do Warburg is to produce bricks for biomass and to increase reductive power in these cancer cells. So many laboratories have been studying the why, no one really understood the how. And it was accepted that some of these cancer cells randomly acquired this metabolic adaptation, and these were selected for. Our results suggested that this may be driven by an epigenetic change. So we decided to investigate that hypothesis that maybe tumor cells get rid of CIR6 to acquire this metabolic advantage. In other words, CIR6 may be acting as a tumor suppressor. And long story short, this is exactly the case. We've been publishing on many different models. Here are two colorectal cancer and pancreatic cancer models of cancer. The moment we delete CIR6, the animals acquire much more tumors, bigger tumors, and in the context of colorectal cancer, we converted normal benign adenomas into aggressive infiltrating adenocarcinomas. In the context of PDAC, pancreatic cancer, we make the disease highly metastatic, something that you don't see in the classical models of pancreatic cancer. So getting rid of this chromatin factor made those tumors extremely more aggressive, and we believe this is true because of a driving in this metabolic change, making those tumors much more glycolytic. Now, we published this few years back. The question that intrigued us at that point, where after we published these papers, was when is the Warburg effect established? And that's what I will tell you in these next few minutes. Again, most of the people in the field of cancer metabolism believe that this was a late adaptation. Rapidly proliferating cells that are in need of doing Warburg for biomass, while they are acquiring more mutations and becoming more proliferative, they need to acquire this adaptation to proliferate. So it was considered that stage 3, stage 4 cancer were mostly doing Warburg. We have some data that suggested to the contrary, that this may be an adaptation that is established very early during the process of transformation. For instance, if you look at the G60 NCI dataset of RNA expression, you can see here the data for pancreatic cancer and colorectal cancer, and you compare a normal tissue to carcinomas, and you see a clear decrease in CIR6 levels accompanied by the increase in the glycolytic genes. In colorectal cancer, we can look at stages. And we saw nicely that even at stage 1 colorectal cancer and in adenomas even, CIR6 levels was already low, glycolytic genes were already high. This was not a late adaptation of rapidly proliferating cells. So we decided to ask in colorectal cancer when during the process of transformation this glycolytic switch happened. And my postdoc at that time, Carlos Sebastian, he's now a professor in Barcelona, asked, so we know that stem cells, normal stem cells are highly glycolytic. So he said, Raul, what about the tumor initiating stem cells? Maybe they're already carrying some of this metabolic adaptation. And actually the intestine is a very unique system to look at this. You're probably familiar with the original description of Hans Kleber of the intestinal stem cells sitting in the crypt of the intestine. These are LGR5 positive cells. What you may not know is that if you eliminate the APC tumor suppressor, from these LGR5 cells, you get full-blown adenocarcinoma. If you do that from the transitional amplifying cells, you get nothing. So Hans Kleber defined the tumor-initiating stem cells in the intestine as cells that are derived from the normal intestinal stem cells. So we have a perfect system. We said, let's look on whether the cancer-initiating stem cells are already glycolytic. To do that, we needed to first define as a control metabolism in the normal intestinal stem cells. And so we look at our conditional CIR6 animals, and to our surprise, already in the normal cells, these are the crypts of the animals, and the CIR6 deficient in the intestine were increasing the normal intestinal stem cells in these crypts. Higher number when you don't have CIR6, and if we inhibit glycolysis by giving dichloroacetate, we saw a major rescue of this increase, suggesting that pushing glycolysis was increasing the number of these normal intestinal stem cells. We were surprised. We decided to test this in organoids, and we saw the same phenotype. For this, we generated a reporter where we put the promoter of PDK1, one of the glycolytic genes I mentioned, followed by the M-cherry reporter, and we saw beautifully rare cells in the intestinal organoids lighting up with the reporter. And we confirmed this by using a staining with an antibody against the phosphorylated form of PDH, the substrate of PDK1. And we got exactly the same staining. So rare cells are lighting up with this glycolytic reporter. So we were sure that this will be the intestinal stem cells, LGR5 positive cells. Now, these cells, as opposed to other somatic stem cells, are rapidly proliferating. And Hans Kleber showed it very nicely. So we thought maybe that's a reason why these normal stem cells are doing wormwood. And this came as a surprise when we stained with both a PDK1 cherry reporter and a marker of proliferation. We saw mutually exclusive staining in the organoids here, where the red cells that are glycolytic are not dividing, and vice versa, suggesting that whatever is this population that is glycolytic is not the LGR5 dividing cells. We were very surprised by this result, and we said maybe this is a weird phenomenon in the normal cells. Let's look at the colon cancer. So we took an APC transformed organoids, we put the reporter infected with the PDK cherry, we injected into mice, and we look at tumors, and we got in the tumors exactly the same pattern. First of all, extremely heterogeneous staining for the PDK cherry, and those cells that were doing glycolysis were not dividing in the tumors. So at least in the context of colon cancer, wormwood was not happening in the rapidly dividing cells. So what were the cells that were glycolytic in the normal intestine? So we went back to the literature. There is a population called the +4 stem cells. These are quiescent, differentiated enteroendocrine cells. They are fully differentiated, but if you eliminate the LGR5 cells, then these cells have the capability to de-differentiate, become stem cells, and populate the intestine. So we asked, since they were quiescent, maybe these were the cells that were highly glycolytic. And indeed, this was the case. We stained with a marker from these enteroendocrine cells, chromogramin A, and we saw perfect co-staining with the phospho-PDH antibody. And lastly, we were asked by reviewers, well, you didn't prove that these highly glycolytic cells, if they are quiescent, are they driving tumorogenesis? So we sorted the cells out of the mice, and we divided the sorting by M-cherry high and low. All the M-cherry low cells, even up to 1 million cells injected, never gave us tumors. And if we injected the M-cherry high positive cells, as low as 10,000 cells were already enough to drive tumorogenesis in these animals. So why quiescent cells are doing wormwood? We follow this, not, I would say, one result that suggests why these cells are doing wormwood is actually to reduce reactive oxygen species. You have here one of these phospho-PDH cells with a marker for reactive oxygen species surrounded by neighboring cells. The neighboring cells show more ROS, and we quantify this, suggesting that the reason some of these quiescent cells, both in the normal and in the context of tumors, are doing wormwood is to protect them from reactive oxygen species. So this is what we show in the intestinal model. We believe that these +4 quiescent cells are glycolytic to reduce mitochondrial respiration, reduce ROS production, and since they are quiescent, these protect them, protect their genome that is sitting there for a long time. The problem is that one of these cells acquire a mutation, for instance, in APC, they may be coming with a head start, metabolically speaking, and that's why we believe we see more tumors derived from the CYR6 deficient animals that are highly glycolytic. And the one message I want to give you from this part is the highly heterogeneous feature of metabolism in this case. Most of the people studying cancer metabolism, they take chunks of cancer and they use this chunk of cancer to do metabolomics and determine how metabolism look in cancer. You cannot do that if metabolism is an heterogeneous feature of cancer. So we need to come up with creative ways to study metabolism if metabolism is an heterogeneous feature in cancer. And I will come back in 2 slides to that. So in parallel, a year ago, we published another paper in squamous cell carcinoma with basically the same results. The only thing I will mention, so you can see here, GLUT1 as a marker of the glycolytic cells is only expressed in the basal cells of the squamous cell carcinoma cells. The difference from colon is here the cells are rapidly dividing. So here glycolysis or Warburg is required for biomass as well. And the CIR6 knockout cells have more of these tumors, bigger tumors, and the unique cells that are affected by glycolysis are these tumor propagating cells with a 4-fold, almost 3-fold increase in these tumor propagating cells in the knockout animals. Interesting, this heterogeneous feature is not only from the glycolytic side. You can see here the marker for histone K56 acetylation, which is a target of CIR6. We see an increase of K56 specifically in the basal cells of the tumor, again pointing to an heterogeneous feature of tumor metabolism. We did metabolomics, we confirmed by RNA-seq metabolomics in vitro, and we confirmed by RNA-seq in the cells the clear signature of stemness, glycolysis, and increase in pentose phosphate to protect these cells from reactive oxygen species. Now, reviewers for this paper did ask us to confirm that these very rare stem cells were doing glycolysis. Again, you cannot do mass spec for metabolomics, but we were lucky to collaborate with Natalie Agar from the Brigham and Women's Hospital. She's one of the few people in the world that can do what is called MALDI-MSI, mass spectrometric imaging, which basically you quantify per pixel metabolite concentration. It's still low resolution, but we managed to do that in our tumors. And you can see here in one slide, you can stain with your marker for the tumor propagating cells. In the neighboring slide, you do metabolomics with this model. And we were able to show that actually reduced glutathione was clearly increasing the tumor propagating cells compared to the non-tumor propagating cells. So we managed to perform almost a single cell resolution mass spectrometry imaging, which I think is where the future of the field will go. So I have a few minutes for the second story. And I did mention briefly that we generated this pancreatic cancer model where when we deleted CIR6, the animal developed this very aggressive metastatic disease. And this is with only the KRAS alone oncogene mutated. Most of the KRAS alone never give you tumors. If we take out CIR6, highly aggressive disease, 50% of these animals with metastasis. And the same is true even in the context of KRAS P53. We are able to push the disease to the left much faster development of tumors. And again, 50% of these animals with metastatic disease. At the time we were publishing this paper, a couple of stories came out from one of the first analyses comparing the genome of primary and metastatic disease in pancreatic cancer. And in both papers that came back to back show very nicely that if you compare the primary tumors to the metastatic disease, there was very little increase in gene drivers, additional gene drivers compared to what you already see in the primary tumors. And actually the paper from Andrew Feinberg went one step further to say, not only we don't see much changes genetically, the main changes, non-genetic changes that we see are epigenome reprogramming of metabolic genes. So this was right out our alley. And when we saw these studies that were correlative in humans, we said we can use our mouse model to try to understand what are these epigenetic changes that happen in metastasis compared to the primary tumors. And we decided to go with 2 different models to expand our horizons. We did both in pancreatic cancer and in breast cancer in a model of 41 cells that you inject into the mice, and in weeks you develop lung metastasis. And because the animals develop metastasis so fast, we hypothesized that they will not have time to acquire new genetic mutations, but rather they should be adapting to grow in this new niche by acquiring non-genetic adaptations. So we did RNA-seq from all these samples, compared the RNA-seq, and we got what we call a metastatic signature. And this was 136 genes that were changing up and down, commonly to all the different metastatic lesions. And long story short, and up to here, many labs have been doing this. We follow with one step further, which was we took the 94 upregulated genes from this group, and we did a functional soft agar assay, high throughput assay in soft agar, where each well we eliminated one of these 94 genes by using an shRNA virus. And in each one of these wells, we tested which of these genes that were upregulated, when we inhibit them, they cause changes in growth of these soft agar colonies. And by using this method, we identified a few genes, a dozen, that gave us clear inhibition of growth in soft agar, while at the same time did not affect expression of 2D growth or the growth of the primary cancer cells. And I will just tell you briefly and rapidly, so we leave some time for questions. We follow one of the genes here called GSTT1 that you can see here in the list of the most commonly affected genes and functioning in our shRNA screening. This is a glutathione transferase, and a very rare one. It's only expressed in the liver, not expressed in the rest of the tissues. And it works different from other enzymes in the glutathione pathway that is used to produce reduced glutathione. GSTT1 is one of the transferases that conjugate glutathione into metabolite and proteins. Okay, it's a conjugate enzyme. So you modify proteins and metabolites by putting a glutathione modifier on those, and it's used in the liver to detoxify xenobiotics. Very little known outside of the liver, no roles in cancer or metastasis for this gene. And we'll just rapidly show you some of our functional data. This is from the mouse PDAC samples and from human rapid autopsy program samples. You see basically in green, no expression of GSTT1 in the primary samples, massive upregulation in the metastasis. Here is by Western. These are few samples from patients from the rapid autopsy program here at Mass General Hospital, and you see massive upregulation of GSTT1 in the metastatic cells. The validation for the high throughput, if we did 2D growth, we see no effect for downregulating GSTT1, but complete disappearance of the colonies. And the same is true in vivo. If we inject these cells in this pancreatic cancer, we created cell lines from the metastatic cells. We grow them by injecting into the mice. The primary tumors, no effect by knocking down GSTT1. The metastatic lesions completely disappear. This is a massive metastatic-specific phenotype. Furthermore, if we overexpress GSTT1 in these cells, we start seeing metastasis in ectopic areas. And you can see here tumors in the diaphragm, shoulder mass, skull mass, when you never see this in other models of PDAC. So overexpressing GSTT1 is enough to making metastasis in ectopic areas. I don't have time to go through these are some data which cells are. This is highly heterogeneous, same as I told you before. For the Warburg effect, we see this GSTT1 expressed in some rare cells. I will not have time to show you why, but we try to understand what is unique in these cells that are GSTT1 positive. We generated an endogenous M-cherry allele to sort these cells out. We did RNA-seq, and we saw clear increase in biological addition and cell addition molecules upregulated, decrease in cell division consistent with these cells being slowly cycling. And lastly, we try to understand what is GSTT1 regulating, and we perform a couple of mass spectrometry elegant samples analysis where we either IP for GSTT1 or glutathione enriched proteins. We identify a common set of proteins, and among them were several extracellular matrix proteins, including fibronectin-1, that were glutathione-related by GSTT1. And if we downregulated FN1, we saw disappearance of these metastatic lesions in the lung from the GSTT1 overexpressing samples, suggesting that indeed the way GSTT1 is driving metastasis is by modulating modification of fibronectin-1. So this is where we are with this story. It was finishing, the paper is submitted, and we will see how it goes, but we believe we have identified unique modulators of metastatic disease that are upregulated in an epigenetic manner. And we believe this gives these cells an ability to grow in this new foreign niche. And with this, I will stop. I think I am right on time. These are all the people in my laboratory. I think, I have a nice picture that are currently in my lab. And this was mostly the work of Cristina Ferrer that already moved on. She's a professor now in faculty in the University of Maryland, and Carlos Sebastian, who is now in the University of Barcelona. And these are the current people in the lab that we didn't have time to tell you about their amazing work. But with this, I will stop and take questions.
Hi, Raul. Yeah, can you hear me?
Perfect. Yes.
Hi, Raul. So you nicely show the phenotype of deleting SIRT6 and what it does to cancer, and then they become more metastatic and more aggressive. So I wonder, what about the converse? Say, can you specifically activate or transgenically express more SIRT6, and what happens to the tumor?
Yeah, so depending who you ask, Chaim Cohen did this work. He created the transgenic SIRT6 animals, and he published a big paper in Nature about extension of lifespan by overexpressing SIRT6. I had a long argument with Chaim to try to see how much of that phenotype was actually protecting these mice from tumors. And we are still arguing about it. He tells me that he believed he see less tumors, but not enough to have statistical significance. And he believed that the reason these animals are living longer are not related to cancer. I believe that a lot of the effect of this overexpressing SIRT6 had to do with the protection of against developing of cancer. But probably you need to analyze much more animals for confirming that phenotype.
So a related question is, do you think activation of sirtuin is a general protective mechanism against cancer?
General sirtuin is activating, definitely SIRT6 I think will help. Activating SIRT6 probably will help. I don't know about other. Many of these sirtuins are showing, hundreds of papers showing tumor suppressive effect for several of these sirtuins. So probably activating other sirtuins may also have a role, but I don't work on the others, so I can comment only on SIRT6.
Thank you.
Great. And while we're on that topic, there's a question in the Q&A about SIRT6 from Larissa Traxler. How is the binding of SIRT6 on the chromatin regulated?
That's an excellent question, and I can tell you that at least in normal cells, we haven't explored this in the cancer context. In normal cells, in the 2010 paper we published, we believe it's brought down as many other repressors by the transcriptional factor itself. We show that in the HIF targets genes, SIRT6 binds HIF1 alpha and is recruited by HIF1 alpha. If we knock down HIF1 alpha, we see SIRT6 going out. In the MYC targets, I didn't have time to talk about MYC co-repression by SIRT6. We see many MYC targets also co-repressed by SIRT6. If we eliminate MYC, SIRT6 is fly out. So there are many examples where the transcriptional co-repressor is brought to chromatin by the transcriptional factor as a way to modulate expression. And we believe this is how SIRT6 is reaching its chromatin targets. We haven't looked in the context of the cancer cell for it, but that's a good question.
And there's a second question that's came right after that that's related from Alexei Anufriv. Excellent talk. And it's a general question. Are there cancers driven exclusively by epigenetic changes?
Thank you, Alexei. You mean, I mean, the question is referring to metastatic disease, whether metastatic disease is, because we know cancers in general, of course, are driven by genetic changes and associated epigenetic change. I don't want to say that only epigenetic changes drives metastatic disease. And we know that in many cases of people has done inducible deletion of oncogenes, you eliminate some of these metastatic lesions. So I think the original drivers that drove the primary cancers are required to sustain metastatic disease. But at least from what is coming from genomic analysis in many new analysis of primary and metastatic disease, people don't see other genetic drivers coming up. So I'm tempted to say that it's mostly epigenetic changes that are driving the ability of these cells to grow in the new niches.
Great, thank you. So I have a technical question, actually. I was really interested. So the MALDI MSI platform is amazing. So what is the, so you have beautiful data. What is the what is the capacity of this? So can you look at multiple metabolites? Is it targeted? Can you just get a small number, or can you give us a feel for it a little more?
So at this point, it's still, I would definitely refer you to talk to Natalie more about it. It's still at its infancy, I would say. The data that I showed was, we were lucky to get enough quantitative difference for reduced glutathione. Many metabolites don't show up. We try to look at all the glycolytic genes and TCA cycle genes, and many of these we did not get enough resolution to look at them. The field is moving very fast. Also, when you look by pixel, it's not really cellular resolution. It's 20-25 microns right now. The new equipment that the Bruker is now developing, they will be even subcellular resolution for much more metabolites. So I think the field is moving there. So definitely it's worth keeping an eye because we will be able probably by using MALDI-MSI to get subcellular resolution for many other metabolites. Still is targeted, but we are moving the right direction.
That's great. It looks amazing. Peter, do you want to jump in? Peter Adams?
Yeah. Hi, Raul. Fantastic talk. That's very exciting. So this function for GSTT1.
Yes.
So can you tell us a bit more? What do you think the glutathionylation of fibronectin is doing? And do you think it has any control in? Does it have a redox control, which is also implicated in tumor genesis, or do you think?
It's
okay,
a good question.
sorry.
We don't believe it's redox. These enzymes don't participate in redox control. And the only function for GSTT1 is in the liver, and it's only on detoxifying molecules by adding glutathione as a conjugate moiety. They don't function in redox. Although it's obvious to think about it because of all the work from Sean Morris on redox control in metastatic cells. We don't believe this is the case for GSTT1. We don't see fibronectin as the only molecule being glutathionellated. We follow that because we need to choose one or a couple.
Right.
Follow. We see several extracellular matrix proteins being glutathionellated. So what we believe is going on from these cancer cells, the GSTT1 is glutathionellating several of these extracellular matrix proteins that are secreted from the cancer cells. This is unusual because most of the people believe that the extracellular matrix is controlled by the fibroblasts, the cancer-associated fibroblasts or CAFs. In our model, we see clearly secretion of these proteins from the cancer cells, which for us was surprising. First, and second, we believe these are mostly non-dividing cells that we see expressing GSTT1. So we believe the cells are sitting there, secreting these proteins to arrange the extracellular matrix in a way that support these disseminated tumor cells to establish in the new niche. Once they are there, and this is a question I got from Bob Weinberg when I present this in a meeting that he was there. I said, Raul, the most important question there is not how these DTCs, the disseminated cells, are there. How do they awaken? How do they start proliferating, which is the question in the field? And I say, yeah, Bob, I wish I knew how they awaken. We still don't know what makes these cells starting proliferating, but at least we believe we define a new mechanism for what allows these disseminated tumor cells to reach the niche.
So does a tumor cell matrix tend to have a higher level of glutathionylation than a normal matrix?
That's what we see in our paper. It was not studied before. No one looked at modification of fibronectin. We started to look, and we do see increasing glutathionate fibronectin 1 in our model. So we believe that may be one thing that is making those ECM proteins better into generating the ECM.
Okay, that's really interesting. Thanks.
Great. And Vera Gorbanova, you have a question?
Yes. Great talk, Raul. Thank you. Well, I thought I'll ask you a question because I get this question many times. I want to see what you would answer to that when I talk about SIRT6 and its tumor suppressive role. That people would often ask me that, well, in particular types of cancer, actually SIRT6 is overexpressed. So can you clarify it for me? What is it actually doing?
I thought you were going to ask me about the DNA repair phenotype, how much it contributes, but before the overexpression, not many cancers. The only one, if you look at the TCGA data, there is one type of tumor that overexpresses SIRT6 as opposed to see deletion. In most of the tumors, we see downregulation of SIRT6, but there is one exception, which is hemato-oncology among malignancy. Leukemias and lymphomas are clearly overexpressing SIRT6. And indeed, there is one paper by Ken Anderson and Dana Farber that show what he believes is the reason. We don't see changes in lymphoid cells for metabolism if you modulate CIL6, at least not in our hands. What we do see is clearly changes in genomic instability from the DNA repair functions of CIL6, and we believe that those tumors benefit from upregulating CIL6 because they become protected against chemotherapy. And Ken Anderson did a very nice experiment where he deleted CIL6 in those tumors. He saw a major increase in sensitivity against the chemotherapeutic agents. So that could be one reason on tumors that may select to overexpress CIL6 to protect these tumors against genomic instability. In most of the other tumors that we look, it's deleted, and we believe it relates to the ability to change metabolism. And to the second part that I know you didn't ask, but I do get this question on DNA damage and repair. I don't think it's only metabolism. It's clear that CIL6 is a key modulator of DNA damage. You show it, we show it, and I believe a lot of these tumors do accumulate genomic instability by not having CIL6. But that just, so it's an additional function.
All right. Thanks so much, Raul. That was a great presentation, great discussion afterwards. So I think
Thank
we
you.
need to move on to the next talk. So we have